Our next speaker is Connor. And what I want to say about Connor is that a part of my job is not only to select the students, but also to find the right match with the mentor. And there are some matches that are really made in heaven, like the one of Connor with his mentor. What I found out totally randomly is that both Connor and his mentor are competitive golf players, like they are in the team at high school. And actually, you think, who cares about golf? But I really feel that the discipline that Connor has with playing golf, precision, grit, really help him in succeeding at this very hard project.
So hello, as you heard, my name is Connor Johnston, and I'm a rising senior at Jupiter High School and was the summer intern at the Fitzpatrick Lab. My project involves investigating underlying structural correlates of functional diversity in the visual cortex, which I know at this point in time sounds like a whole lot of jargon, but in a couple of minutes I promise it will all make sense. So the primary visual cortex is a key area in our processing of visual stimuli. When we observe something with our eyes, our retinas send information along the optic nerve to our visual cortex, a similar path to what you see here. In the visual cortex, neurons then encode images into lines and edges to help us recognize the world around us. And beyond this, we actually know very little, hence our focus on mapping and modeling the visual system. So our lab studies two species, tree shrews and ferrets, and for my project, I work with data from the former. Tree shrews contain a very well-developed visual system, often considered to be one of the more complex amongst animals. As extremely visual creatures, most of their information intake occurs through the visual system, resulting in a neural anatomy akin to that of higher mammals, including humans. So working with tree shrews allows us to work with squirrel-sized animals that are easy to care for and handle, while also yielding findings that are generalizable to ourselves. So within the primary visual cortex of tree shrews, neurons exhibit orientation tuning. Orientation tuning is simply a neuron's preference to the orientation of a stimulus, specifically lines or edges. And this video was gathered from 2-photon imaging of a living brain in combination with an activity sensor that lights up when a neuron is excited, known as GCamp. And in the top left corner, you can see what the animal would be seeing. And I'd like you to notice that as the orientation of these flashing lines changes, different regions of our brain lights up, which, when we color code, can show us how neurons organize themselves according to their preference and surround themselves with similarly tuned cells. This is referred to as modular organization. Now, this firing of different areas is, in real time, what actually happens inside our brains as we visualize the world around us. And we can quantify this response for each and every neuron and graph them like seen here, which just goes to show how selective these neurons are for their particular stimulus. And this brings us to the concept of orientation clusters. Orientation clusters, simply put, are groups of neurons that share orientation tuning properties. So each of these colored regions are orientation clusters. However, within orientation clusters, cells can have different preferences to the size of a stimulus. This is referred to as size tuning. My project is centered around two functional types of size tuning, summation and suppression. To understand the differences between the two, we should look at a quantification of the output from a cell of each of these functional types. The scales of these graphs show the neuron's output on the y-axis and the size of the stimulus on the x-axis. And if we trace these graphs in tandem with an example stimulus, we can see the correlation in each graph between the output and stimulus size. So what is causing this difference in output in two cells that are tuned to the same orientation and within the same orientation cluster and anatomically very proximal to one another within the brain? So to understand the output of a neuron, we first have to understand the input. We've always known neurons to consist of the dendrites, the soma, and the axon, with information flowing through them in that order. As such, we have to look at the dendrites to comprehend input. The dendrite consists of two domains, the apical and the basal arbors. In figures, apical will always be above and basal will always be below, as this is how they orient themselves within the actual brain. And I'd like you to remember back to that rainbow video I showed you a couple slides ago. In each region you watched light up with GCamp was purely the soma, or this small red dot here. In reality, each of those lighting up regions has a field of dendrites very similar to this, which just yields a remarkable level of complexity within our visual cortex. As previously mentioned, the dendrites are the site of incoming signals. They form connections with surrounding neurons known as synapses via small protrusions called spines, which you can see here in green. The number of spines helps us to roughly determine the number of information inputs along a cell. So, for my project, I studied the dendritic morphology of a sample of layer 2-3 summating and suppressed cells from tree shrews in hopes to quantify a structural difference between the two ends of this functional diversity spectrum. We don't quite have an understanding of how a cell's morphology relates to its functional response, so this was our logical first question and first step. To answer this question, we first introduced an activity sensor called GCamp into V1 neurons, which is the same GCamp that you watched light up in the first video. A visual stimulus was then shown to the subject with varying size, and activity is tracked with each stimulus to determine functional type. Again, either summation or suppression. The use of GCamp is referred to as a sparse labeling method, as it would tag entire orientation clusters with fluorescence, allowing us to look at fine morphological details while not having our target cells occluded by other neurons. The treated brains were then mounted and sliced into 50-micron thick slices. And for reference, this is about half the width of a piece of traditional printer paper. These slices were treated once more with an antibody to promote fluorescence, and then the images were scanned with a confocal microscope and run through the neuromorph program, which started a semi-automated reconstruction process, which would later be finished by myself. So I was involved in the slicing until the complete reconstruction of the neuron, whereas postdoc researchers performed the fluorescence and showing of the visual stimulus. So this figure best encapsulates the pipeline of how we take serial slices and turn them into complete neurons. On the left, you can see our tissue slices, slice 3 and slice 5 respectively, each 50 microns thick. As such, this top row shows the part of our cells that are 150 microns below the cortical surface and will be our apical arbors, whereas this bottom row shows the part of our cells 250 microns below the cortical surface, or our basal arbors. This middle column is the result of our confocal imaging, which, for those curious, is a 20 times zoom of these wide field images of our slices. And this final column shows the outcome of neuromorph and manual editing, which is a complete skeleton of that layer's dendrites. The aforementioned slices and their skeletons are aligned in a three-dimensional space, and as you may have noticed here, there is quite a lot of extra dendrite, including an entire extra cell, which is the main drawback of our sparse labeling technique, as it may not often be as sparse as we would hope it to be. So our applicable dendrites are spliced together, and extras deleted and removed, and this yields complete reconstructions of our neurons. So these are the dendritic arbors of our 8 sample cells to scale against one another. Our summating cells are in red and our suppressed are in blue. And to draw conclusions from this data, I chose to quantify some of the characteristics of these dendrites. So this is an example from 2 of the cells, summating and suppressed. The first column of graphs shows the dendritic length at cortical depth, and this shows us general dendrite size and the dispersion of dendrites relative to depth. These remaining graphs show branch points at cortical depth, which a branch point is a point of bifurcation along a dendrite, which is where a branch will split into 2 more branches. We use branch points to quantify the complexity of a cell. So a cell with a higher number of branch points is considered to be more complex than one with a lower number of branch points. And when we compared these graphs of all 8 of our dendrites, no trends emerged that would differentiate between the functional types. So this is a plot of that data. We see summating in red, suppressed in blue as per usual. And in total dendritic length, we don't see any trend that would differentiate between the functional types. So I plotted the data for each dendritic domain, which we begin seeing some interesting dispersion within a functional type, but again, nothing that would differentiate. So if we look at the total branch points, again, we see no clear differentiation emerge, which holds true when we plot for the domains. At this point, we have extremely high-resolution images of our cells and complete dendrite reconstructions, so we chose to look at spines. So spines have to be manually mapped due to accuracy concerns, and to do so, I would go layer by layer, manually selecting each individual spine. And we gathered spine data for 6 cells, and that gathering process looks something like this. And of those 6 cells, we gathered 3 of each functional type, and in total, I mapped over 10,000 individual spines. These spine maps would then be aligned alongside the dendrite models, yielding a complete reconstruction of our dendritic arbors. So this is one example of the spines we mapped for the sample cells, and images like this exist for all of the cells. And by zooming in, we can see this marvelous complexity of spines and how their orientation changes along the dendrites. And these spine maps can be used to calculate any measure of spine-based data, as well as for studying functional architecture. We chose to look at spine density as our medium of comparison, having calculated the total number of spines for each of our cells, but attributing that to a difference in cell size. So this plot follows the same rules as the previous one, summating cells in red and suppressed in blue. And we can see that spine density for both functional types are very similar. And when we plot that for each domain, we see some interesting grouping amongst a functional type, but we don't see anything that would help differentiate between the functional types. So as seen, there was no clear correlation found between functional type and morphology. However, the sample size was relatively small. Gathering more data over a longer period of time may allow for some regularity to emerge within the population. And a cell's morphology tells us how a cell samples from its surrounding environment, but there could be more directed input that is not exactly reflected in this sampling strategy. Therefore, we may want to stain for inhibitory inputs along the dendrite and the soma and look at functional properties of the cells in the network in which our target cell is embedded, and perform functional spine imaging to observe the actual influence of these excitatory networks on our cell. If we can map the function of these inputs overlaid with the morphological data that I've gathered, in understanding the distribution of these types of inputs, we can actually determine the functional architecture of our cells and hopefully find an explanation for the difference in functional type. I would like to thank Dr. Fitzpatrick for welcoming me into his lab for these 6 weeks, and to Ilaria and Erica for coordinating this wonderful opportunity for myself and fellow interns. And I'd like to extend a special thank you to Drs. Mike Guest and Jad Barshini for being wonderful mentors for myself and teaching me everything I know and including me in this amazingly fascinating research project. And thank you to all the wonderful MPFI donors who make this program possible, and to the Fitzpatrick Lab as a whole for supporting my learning throughout this internship. I'd now like to open the floor to any questions.
Questions for Connor?
Thanks a lot, Connor, for a great presentation. Here, early in your presentation, you talked about the orientation of the dendrites, and almost like a north-south orientation. Is that true in the visual cortex, or did I just misinterpret what you were putting on the screen?
So the question is the orientation of our dendritic arbors holding true to what I said in the visual cortex? So it is the case, yes. The apical arbor of our dendrites will always point towards the surface of the brain, whereas the basal will always point towards the bottom. So as a general rule of thumb in figures, we cast the apical above the basal. So maybe not quite a north and south orientation as you implied, but maybe an out to in. Oh, of course, thank you for the question.
Any other question?
I'm just curious, is there any work in your lab about the plasticity of the neurons, meaning like will dendrites grow if you like activate them, like give the same stimulus over and over? Or if some parts of the brain stop working, will the dendrites change? I'm just curious.
So the question is, are there any research studies in my lab surrounding neuronal plasticity? And there is some work in my lab. My lab on the website encapsulates 4 projects. My project that I was involved in has to do with visual stimuli and how that affects dendrites. But I'd be welcome to talk with you after with my mentors about the specific projects that are occurring about neuronal plasticity. Or if you're curious, there is the website that does go much more into depth about those.
I have a question for you.
Mm-hmm.
Did you ever thought about quitting between the spine number 1 and the spine number 10,000?
No, it was honestly very therapeutic to sit and click on the spines.
Okay, thank you so much.
Thank you.
Great job.